In this video, we're going to look at the rhetorical patterns of introductions in Ames pages. This is the first of a two-part video sequence on Ames pages. This video, the rhetorical patterns of introductions in Ames pages, is going to focus on the typical patterns you see in the introductory material. And we're going to deconstruct two examples to show how those examples share similar rhetorical patterns. In the second video, Patterns in Content in Ames and Conclusion in Ames pages, will focus on the Ames themselves and the concluding content of the Ames pages using one of the examples from this video. Let's look at the rhetorical patterns of an Ames page example from a funded NIH grant. You can download a copy of this Ames page from the Climb website at the link below. So let's look at the example. Here we're going to see an opening paragraph that funnels to a complication. Eukaryotic innate immune systems act as effective barriers to infection by microorganisms. Understanding the mechanisms that bacterial pathogens employ to circumvent innate immune systems will improve our ability to control disease. Now, this opening sentence establishes a general context, and it also establishes the significance of this research effort. Next, we move to a narrowing context. Plant and animals use specific pattern recognition receptors, PRRs, to recognize conserved molecules of microorganisms, known as PAMPs. Plants have numerous PRRs that can recognize specific virulence proteins specifically present in pathogens. Many gram-negative bacteria use type 3 protein secretion systems to inject effector proteins into host eukaryotic cells. So we've gone from the general context to a narrowing context, focusing on the research effort itself. And next we have an interesting sentence, which underscores the research contribution of this researcher. We have shown that a primary role for many Pseudomonas syringae type 3 effectors is to suppress innate immunity. And finally, we conclude with the complication and a great closing sentence that harkens back to that original significance. However, the enzymatic activities and the mechanisms that type 3 effectors use to suppress innate immunity are not well understood. Identifying the enzymatic activities of type 3 effectors and their substrates is essential to identify important components of innate immunity and to improve strategies to control bacterial diseases. As you can see, the complication is signaled by that typical word, however. And that opening paragraph funnels from that broad, general context into a narrowing context into a complication. Now, often you might see writers and speakers move directly to the hypothesis, but that's not what happens here. Here instead we see a rhetorical move that focuses on long-term and short-term research goals. Let's take a look. First, we have the long-term goal of the research. Our long-term goal is to elucidate the molecular basis for suppression of innate immunity by type 3 effectors. This is the long-term objective of the research, not this specific application, but where the researcher wants to go down the road. Next, we have the specific goal of this research application. The objective of this application is to identify targets of the P. syringae type 3 effector HOPU1, a mono-ADP ribosyltransferase, and to determine its role in bacterial pathogenesis. So you see, we moved from a long-term goal to a very specific goal of this research application. So after we state the hypothesis, we see two additional rhetorical moves. First, a summary of the research, and second, a series of sentences that stresses the qualifications of this research team. First, we explicitly state the hypothesis. The central hypothesis of the proposed experiments is that the targets of the HOPU1 ADP-RT type 3 effector will be components of innate immunity. And then we shift to a summary of the research, which provides a path to the hypothesis. And here we want to stress the word summary. We formulated this hypothesis based on the literature and on our research on other type 3 effectors, as well as our preliminary data showing that HOPU1 suppresses outputs of innate immunity. Recently, we have shown that HOPU1 can use several Arabidopsis RNA binding proteins as high affinity substrates in vitro ADP-RT assays. Based on our preliminary data, one of these proteins, ATG-RP7, plays a role in innate immunity. Notice how this discussion deals with both research done by other research teams as well as the research and the preliminary data acquired by this research team. Next, we have an addition to this specific research goal. A major goal of this application is to elucidate the function of this protein as it relates to innate immunity. And then finally, we have a series of sentences that stresses the qualifications of this specific research team. So now you see all the rhetorical components of these two introductory paragraphs in this Ames page. Let's look at the introduction pattern of another funded NIH proposal. You can download a copy of this Ames page from the CLIMB website at the link below. Now, this example takes a somewhat different approach. It immediately gets into the long-term goal of the research and then moves into the general context, which establishes the significance and articulates a general complication. Let's read this opening paragraph. The long-term goal of this research project is to identify the optimal dose and schedule of administration of drugs active against influenza viruses that will prevent and or cure people with influenza without causing the emergence of resistant viruses. The adamantines and neuraminidase inhibitors have been used for the prevention and or treatment of influenza. However, they often fail because treatment with these drugs leads to the emergence of resistant viruses in the treated population. Notice how all three components are kind of woven together in one paragraph. The next paragraph starts digging deeper, narrowing both the context and the specific complication. Adamantines have historically been used in the treatment and prevention of influenza A virus infections. Recently, viruses that are resistant to these inexpensive drugs have emerged, rendering them less useful for therapy of influenza. Neuraminidase inhibitors represent a new class of agents for use against type A and type B influenza virus infections. While shown to be effective, there have been instances of emergence of resistance or reduced sensitivity during therapy with neuraminidase inhibitors. This has been seen especially in children where high clearances for these agents in general and oseltamivir in specific are the norm. So you notice, in other words, it's moved from general to specific, alternating context complication, context complication. Now the next paragraph takes up the other critical rhetorical components. First, we have a summary of the research that creates a path to the hypothesis. The hollow fiber infection model system has been used to determine the optimal dose and schedule of the administration of antibacterial, antifungal, and antiviral compounds for use in the treatment of individuals infected with bacteria, fungi, and viruses. Next, we have the specific and narrow research goal. We propose to use the HFIM system to study the effects of amantadine and neuraminidase inhibitors, oseltamivir, carboxylate, and on the replication of influenza viruses, and to identify the pharmacodynamically linked variables for these antiviral drugs, alone and in combination, with respect to inhibition of virus replication. We also propose to identify whether a different pharmacodynamically linked variable is present for suppression of emergence of resistance. Then we have our hypothesis. Again, it's explicitly stated. We hypothesize that HFIM system can be used to provide information on resistance selection in humans, and that the HFIM system can be used to determine the dose and administration schedule of antiviral compounds and combinations of antiviral compounds that will inhibit the replication of influenza viruses while preventing the emergence of resistance. And then finally, this concludes with the qualifications. That happens in this instance after the specific aims are stated, but that same component is still there. So let's look at the two rhetorical patterns side by side. Similar components, but different sequence. Example 1 on the left, example 2 is on the right. Though the research subjects are very different and the organization seems just as different, both have the same components. Both have a general context and a narrowing context. Both have complications. Both specify the narrow research goals of the project. Both have a summary of research that establishes a logical path to the hypothesis. The sequence is different, but the parts are almost identical. Commonalities, opening paragraphs, funnel to a complication. The significance is clear. We know why this research is important. There's a long-term goal, a short-term goal. The hypothesis is explicitly stated. We know where it is. The summary of key previous research, we know where that is. And the qualifications are articulated. So your challenge when you are creating an aims page, whether it's for a dissertation proposal or for a grant proposal, is to identify the rhetorical components you need and arrange them logically. To that end, this template may help. First, you need to establish the general context and significance of this research. What's the big picture for the research? Why is it important? Next, you need to have a narrowing context. What's known and accepted in your research area? Next, you might add your research contribution. Has your previous work contributed? How has it done that? Next, you have to have a complication. What's the problem? What's the roadblock? What's the unknown? Next, you have to have a long-term goal. What final big result will this research help achieve? Not today, not tomorrow, but down the road. Next, you need a specific short-term narrow research goal. What is this research going to accomplish specifically? Then you need a summary of the research, a path to the hypothesis. How does the previous research lead to the hypothesis? How does your research, previous research, and preliminary data lead to the hypothesis? Then you need to state the hypothesis explicitly. What do you believe to be the answer to the complication that you've raised? And then finally, you need to stress the qualifications that you have. What makes you the right person to undertake this research? When you understand these rhetorical components and arrange them logically in your aims page, you can put together a powerful and compelling single aims page for the research that you want to undertake.